Bismillah ar-Rahman ar-Rahim. We will start the second session. We have now the privilege to introduce Dr. Thana, with extensive experience, has made significant contributions to the field. Dr. Thana Rifa'i is a consultant and head of a clinical chemistry unit in Amiri Hospital, Ministry of Health in Kuwait, and she is a professor of clinical and chemical pathology in Ain Shams University, Cairo. Dr. Thana is a clinical tutor and site coordinator in Clinical Biochemistry and Metabolic Medicine Board in Kuwait, which is part of the KIMS, Kuwait Institute of Medical Specialization. So her topic today about clinical chemistry. Dr. Thana, please.
Assalamu alaikum wa rahmatullahi wa barakatuh. Good morning, everybody. It is my pleasure to be with you today in this distinguished scientific event. My presentation will be today on the part one, quality control, as part of the session of quality control and quality assurance. During the presentation, we'll go through a simple introduction, relation between quality management, quality assurance, and quality control in clinical laboratories. We'll go in short in covering the quality control programs, mainly today the internal, and tomorrow will be the interlaboratory program and the external one. So our focus today will be on the internal quality control program, going through the control materials, errors, design of the internal quality control program, QC rules, limits, and statistical terms related to QC program, some troubleshooting about rule violation, Six Sigma and QC design, quality control models based on Sigma metrics and independent QC material. Nowadays, you know that more than 70% of medical decisions are based on laboratory data that require to be accurate, reliable, and timely. So we have to give a quality result. So what is quality? Quality means meeting the requirements of the users and customers, and our users are the doctors and nurses, and our customers are the patients. So what is the lab quality? The lab quality, why lab quality? Lab quality is essential for better patient outcome, and most important is to avoid the bad consequence of poor quality, which is inappropriate treatment and follow-up plans, delayed treatment, loss of credentials, and up to legal actions. So we'll go to see some relation between terms related to quality in the laboratory. We have quality management, we have quality assurance, and quality control. The principles of quality management, assurance, and control are the foundation by which clinical laboratories are managed and operated. Lab QMS, or quality management system, refers to a set of policies, procedures, and practices implemented to ensure quality in all laboratory operations. So it is like the umbrella which covers all the policies, procedure, practices in all your laboratory operations. So the TQM, which is called the Total Quality Management System, involves planning, control, and monitoring of all laboratory activities to comply with regulatory requirements, maintain reliable results, and enhance customer satisfaction. So what is quality assurance? Quality assurance, as we said, the total quality management is the umbrella covering coming within it the quality assurance, and within it the quality control. The quality assurance, or called quality assessment, it is a part of the TQM that helps to provide confidence in the quality of the total laboratory testing process. It can be summarized as ensuring the release of right result at the right time, on the right specimen, from the right patient to the right location, with proper interpretation based on reference and date. So by defining this, so the quality assurance involves activities inside and outside the laboratory that involve the, I want to show on the figure, please. Yes, this one. So it entails the whole total laboratory process, pre-analytical, analytical, and post-analytical, from test to order till releasing the result with proper interpretation to the clinician. So what is the main difference between quality assurance and quality control? Quality assurance is process-oriented, unlike the quality control, which is product-oriented. The quality assurance is planned to avoid error, so it is all about prevention. On the other hand, quality control is try to find or to detect errors, correct them, so it is all about detection. Quality assurance involves, as we said, all the phases of the testing process, so it is system-oriented. Unlike the quality control, it is a tool to detect and correct errors, so it is focusing on the analytical phase. And this diagram shows you how the quality management system is the umbrella covering all the process within the lab. Then the quality assurance is concerned with the quality of the total test process, and the quality control come to detect and correct errors in the focusing on the analytical phase. So implementation of quality assessment or quality assurance system for the analytical phase is an essential task, and it's fulfilled by two key activities, the internal and the external quality assessment programs. So we have other indicators or other control indicators for the extra examination phase, which are the pre-analytical and post-analytical, and they are known as KPIs, like turnaround time, specimen rejection rate, patient identification error, and it is out of the scope of our presentation today. Regarding the quality control program or the statistical control of the analytical phase, it is an integral component of quality assurance and an essential element of QMS, as we showed in the figure. And it covers a part of quality assurance which concerns the control of errors in the analytical phase. It's achieved by what? By testing materials of known values to monitor the accuracy and precision of the testing process. And the goal of the quality control, as we said, it is all about detection. So to detect, evaluate, and correct errors in the testing process before analyzing and releasing patient reports. We have, as we said, internal and external quality assessment programs. The external quality assessment program is known as proficiency testing, and we'll talk about it tomorrow, inshallah. Both programs are mandatory by all accreditation programs like ISO 15189, a CAB, a joint commission, to ensure patient results reliability and better outcome. So we'll go through the internal quality control today in some details. So the internal quality control program design, laboratories have to develop their own what's called individualized QC plan, because simply one QC procedure does not fit all lab tests. So this is what we tell, one size does not fit all. So you have to develop your own individualized QC plan. So what are the components of this plan? You have to list all the variables that affect the lab result regarding the analytical process, water quality, pipette accuracy, analyzer performance, calibration, maintenance, and so on. You have also to design or to mention your QC testing protocol and control limits, the methods of storing and evaluating QC results, frequency of running the QC levels, and the procedure of troubleshooting and corrective action. And last but not least is the record retention and documentation. Regarding the QC materials, we have two main types of quality control material, the liquid one and the lyophilized one. Both of them are stable. The liquid is ready for use. It doesn't have any reconstitution error, no lyophilization error, and mixing is not required. Unlike the lyophilized one, which is reliable, have the possibility of having lyophilization error, reconstitution error, and mixing error as well. We have to note during the preparation of our control material is to use the proper volume of distilled water and check its quality, proper calibrated pipettes, assure complete mixing, and the proper identification on liquids. So when you are selecting your internal QC material, you have to notice the following. It should be at or near the medical decision limits and covering the entire analytical range of the analyte. And we faced a problem when we changed it from the conventional troponin assay to the high sensitive, when we were still having the old control for the convention one, which was not covering the low level. So we specially requested, we had a special request to request the CLT levels, which cover the low level. Also, it has to be determined by your lab platform and peer group, and ensure supply of at least 1 year of the same lot number, and to have as little as or zero variation within the same lot number between vial and vial. So we have to go through some basic facts. to allow us to troubleshoot later on the internal QC troubleshooting reports. We have to know in brief the analytical errors, types of analytical errors. We have the random error and we have the systematic error and the total error. The random error is an error that is its direction and magnitude are unpredictable, and it is a measure of imprecision. The systematic error is an error which is consistently low or high, and it is measure of accuracy, also known as bias. It could be constant or proportional. And from this figure, we can see the bias or the systematic error. It is a shift of your mean from the true value. And the random error is numbers of multiples of standard deviation. So it's about the variation of the process or the imprecision. So what are the causes of such errors? If you have random errors, the most frequent causes are related to the instrument, like imprecision in pipetting. Maintenance is required for the photometer, for the reaction cuvettes, for the lamp, temperature instability, improper mixing of the reagents with the samples, bubbles or particles in the reagent, or sample line problem in the automation line. For the systematic error, if you got systematic error, mainly focus on the calibrator, and mostly it is corrected by recalibration. And the reagent also can be source of systematic error, and it can be in the QC material itself. Any one of these, improper reconstitution, it is near the expiry date or expired or deteriorated, or regarding the calibration function was unstable, or a change in the methodology. So now we will shift to the QC charts, limits, and rules. And this is our famous visioning chart, QC chart, which shows, which we are plotting every day our QC data. And on the x-axis, we have the days or the runs. And on the y-axis, we have the mean plus or minus 1, 2, and 3 SD. And the perfect chart or the ideal QC chart is the one with fluctuating around the mean above and down and within the control limits. Regarding the limits and the QC rules, sources of control limits, usually we have the manufacturer limits, which we are getting in the beginning, and they are wide with low error detection probability. We develop our own QC limits using 20 to 30 QC data. It is better because it is narrow and representing the lab performance with higher probability of error detection. And we have with time to develop our cumulative interval, which is the floating interval, using 2 months or more QC data to update the QC limits on monthly basis. Regarding the QC rules, we have single versus multi-rule, and we have statistical versus clinical limits rules, which is represented by the total allowable error. Regarding the single rule, it is simply using one and multiples of standard deviation as rejection rule if violated. It can be 1 to S, 1 2.5 S, 1 3 S. So when it can be used with high precision automated systems with stable performance. And if the single rule is able to detect 90% of the critical errors in a single run. In the 80s, James Westgard proposed alternate control rules to limit the false rejection probability of the single rule, while at the same time maximizing the probability of the error detection through application of multiple rules. And this is our famous traditional Westgard multi-rule, showing 1 to S, 1 3 S, 2 to S, and R for S, 4 1 S, and 10 to the mean. In case of running 2 QC levels, in case of 3 levels, you have other rules. They provide a clue. Advantage of these multi-rules, they provide a clue on the type of analytical error. Is it random or systematic? Because 2 to S, 4 1 S, 10 to the mean rules are more sensitive to systematic error. So when you get one of these, you are directed to the causes of systematic error. On the other hand, 1 3 S, R 4 S are more sensitive to random error. But all of these rules would be described as statistical process control. Some Westgard rules are considered as warning signal, like 1 to S, 4 1 S, and others as rejection or out of control signals. So what is our response to this? Our response to warning signal, it is not just go through the run and wait for the other control to be run. We have to perform maintenance before next run. We have to inspect the analytical system and review system changes. While the response to out of control problems, here, we have good lab practice and we have bad lab practice. The good lab practice is to identify the error, is it random or systematic, so that to direct your correction or fixation of the problem to the causes of these such errors, so relate error to potential causes, and consider factors in common. For example, if you have an out of control for sodium, potassium, and chloride, so focus on the ion selective electrode problems, things in common. Relate causes to recent changes. For example, if you got the out of control, when you changed the reagent load, the calibrator load, or maintenance was done, so direct the causes to the recent changes, and verify the solution and document it. Unlike the bad lab practice, which is unfortunately the most frequently done, is just repeating the QC, trying a new QC, and then calibration without any indication for calibration. And still, the problem will be there if there is no problem in one of these. So regarding the out-of-control data, when you get out of control, what you will deal with it, they have not to be included in further assessment and also not to be included in calculation of your next cumulative mean and standard deviation. We will go through rapidly in some troubleshooting reports for internal QC. This is the leveraging chart from LIS program showing 2 levels of control materials and their performance all over the days. And this is a nice performance because they are all green. So we like the green color in the QC chart. So it is fluctuating around the mean and within the control limits. This is a chart showing a 2 to S rule violation across QC material, and calibration was done and it was corrected. This is a chart showing example of 1,3S when we get in one level, and usually 1,3S denote random error, but also it can be a very early sign of systematic error. But here, when they did the maintenance, it was corrected, so it was a random error. Here another example for random error, which is R4S, when we have across control levels, between the 2 control data, more than 4 SD. So it is also very sensitive to random error. So we will direct our correction toward the instrument. There are limitations, some limitations for this statistical process control multi-rule, that they are not considering the intended medical use of the test, and they rely purely on the statistical criteria. So these limits reflect the analytical performance inherent in the method. And laboratories with different analytical performance used control limits, which did not promote harmonization between laboratories. In the second half of 90s, with the introduction of the concept of quality specifications based on biological variations, the published list of allowable bias, allowable imprecision, and allowable total error, and their impact on patient care, the recommendations are to shift from using statistical QC limits to the medical decision limits. Why? To eliminate statistical rejection, to provide wider control limits that better reflect the clinical use of the analyte, which is of more concern, not the statistical issue, and reduce the false rejection probability, which is coming with the statistical process control. So now, once we talk about the medical decision limits, we have to go through, in short, about the 6 sigma and how they impact the QC design regarding the limits and the frequency of running the QC material based on the sigma metrics. So what is 6 sigma? 6 sigma is a method of process improvement focusing on minimizing the variability through reducing the number of errors with process repetition. So sigma models refer to the number of standard deviations or sigmas that fit within the quality specifications of the process. As shown here in these 2 curves, this with 3 sigma performance and this with 6 sigma. So the one with 3 sigma only fitting 3 standard deviations between the limit, the allowable limit, and the mean and the true value, while the 6 sigma show very little variations where it can accommodate 6 standard deviations within the allowable limit and the true value. So how 6 sigma impact our QC? This table also shows how it shows the defect per million opportunities for the sigma level, where the 6 sigma shows you when you repeat your assay or you repeat your process million time, it only gives 3.4 errors. So that's why it's called world class. So when you repeat your QC performance a million times, you are only getting 3.4 errors if your assay is performing as 6 sigma. In the 21st century, internal QC models based on the analytical performance of the method represented by sigma metrics and total allowable error were developed so that individualized QC plan per analyte and method can be set up. Sigma metrics, how it is calculated for quantitative assays, you calculate according to this equation, where sigma equals total allowable error minus the percent bias divided by the percent CV. From where you get your bias? From your external quality control or inter-laboratory program, as we'll see tomorrow. And also you can get it from during doing the method evaluation in the beginning of adopting the assay. Based on this equation, and the CV, you know, it's coming from our internal QC over the preceding months using this equation. The Six Sigma model allows laboratories to set their QC plan, as we said, and if you don't have enough informatics support to help you in this application, you can refer to this nomogram developed by Westgard to help you in designing your Sigma internal QC run size and the number of QC to be run and the rules to be applied. So here on the x-axis, your sigma metric, and here is your workload. So you define your workload, which is the number of patient samples you will run, and these oblique lines are the QC, the number of QC rules, the number of QC levels to be applied, and the rules to be applied also from up down, as seen in this key box on the right side. Here we simplify the issue when you have sigma metrics, when you have an assay with world class or 6 sigma. So this diagram was done by Westgard. This is a modified Westgard sigma QC rule. Unlike the other one, was not considering the performance of your assay based on sigma metrics. So on the bottom of the chart, you will find your level of sigma. And these dashed arrows, on the left of the dashed arrows, it will show you the number of the suggested number of controls to run, and the run size, and the control rules to be applied. So with six sigma, as shown in the table, you can control your run by doing only 2 quality control levels, and you apply only 1 3S as rejection rule, and you can run the QC every 1,000 patient samples. Unlike the low sigma, you have to increase the number of control levels to be run. Also, you have to apply all the rules, Westgard rules, to be rejection with a little run size. So the higher the sigma, the better the method performance and the more relaxed QC limits, frequency, number, and rules. And this is just to sum up how sigma affected the QC design. For large sigma process, more than or equal to 6 sigma, simple QC rules with low false rejection probabilities are adequate. For intermediate sigma values between 3.5 and 6, so the quality goal can be met, but you need more elaborate QC strategies are required. For low sigma, which is less than 3.5, it will be difficult to meet laboratory quality goal, except if you do changes in your analytical system to reduce errors as much as possible. We will talk in brief about the independent QC material or what is called third-party control. They are stabilized controls independent of the analytical system and are provided by another manufacturer. They are highly recommended by accreditation standards as by BERCAP, ISO 15189, who stated that use of independent third-party control materials should be considered either instead of or in addition to any control material supplied by the manufacturer. CLSI also insisted not to use the same quality control from the same manufacturer who manufactured the calibrator material. So what are the advantages of the independent QC material? They are assigned using a large number of independent laboratories, thus ensuring statistically valid targets. They provide unbiased results and allow detection of analytical test system errors, calibrator, and or reagent issues that are not detected by the manufacturer-dependent QC. They are very sensitive to any shifts in the test system performance, and they monitor the analytical performance throughout the patient reportable range, especially at the clinical decision limits. So now to summarize the present situation of the internal quality control program, we have strong points and we have weak points. We have strong points which is helping us and emphasize our process in the form of presence of well-established internal QC protocols to monitor the analytical performance. Data on biological variation to establish control limits are highly reliable and exist for the majority of analytes. The workload and the impact of an analytical failure on the information concerning patient status is easy to be known and should be applied in the internal QC protocol. And third-party, non-commutable control material exists to produce good internal quality control and to provide information from other laboratories using, which is your peer group, using the same measurement method and same control material. They are non-commutable means that they are not behaving the same as patient sample. So there are efforts now to produce a commutable independent QC material which behaves similar to patient samples, but the production is much less than the market requirements, and its stability also requires to be stored at -80. The weak points to be eliminated about the internal QC situation now is using 1-2S single rule. Because of its high probability of false rejection. You know, the 1-2S has a probability of false rejection. If you run one control, it's 5%. If you run two controls, 9%. If you run three controls, it becomes 14%. Using control limits simply based on statistical rather than control rules derived from quality specifications has to be omitted also. Repeating control tests without searching for the reason of failure. So we have to return to the reason of failure is that control a violation showing random error or systematic error. We have to go and deal with the cause properly. Repeating all patient samples without checking where the failure was produced and releasing patient results when a failure was produced. And we will sum up or finish our QC by this saying of James Westgard: QC is not a tradition, something handed down from a generation to the next. It is a science. So if better control rule does the job, use it. And thank you.
Thank you, Dr. Thana. Following Dr. Thana, it's our honor to present Dr. Heba Kary, visiting us from Saudi Arabia. Dr. Heba graduated from King Abdulaziz Medical School in Jeddah, Saudi Arabia. She's Canadian board certified in medical biochemistry from McGill University. She currently works as a consultant and a chemical pathologist at King Faisal Specialist Hospital and Research Center. She's also a well-known member of the Point of Care Committee of IFCC and AACC Academy Guideline Committee for Reproductive Point of Care. Welcome, Dr. Heba.
Good afternoon, everyone. How are you? Thank you for coming, and I'd like to start by thanking the organizing committee and the scientific committee for the invitation and the hospitality. Today, I'm talking about the role of QC in point-of-care testing, and it's pretty much similar to what Dr. Kotb was talking about previously. It's not that different, but just bear in mind that whoever doing the quality control in point-of-care is actually an end user and they are not laboratory. Before I start with my presentation, I would like to introduce to you the hospital that I work in. It's a new branch for King Faisal Specialist Hospital and Research Center. It's located in the holy city, Al-Madinah Al-Munawwara, where Rasul Sallallahu Alaihi Wasallam buried there. The hospital is newly opened end of 2021. Our scope of service is oncology, ophthalmology, and OB/GYN. The hospital, we managed to open 60% of the bed capacity so far, and we have a total of 212 physicians. And I'm proud to be one of those physicians that is very hardworking and have achieved a lot in a very short period of time. These are, I'm mentioning here a couple of the international awards that the hospital have achieved over the last 2 years. In 2022, the hospital went top 3 project worldwide by Project Management Institute for the large and mega project category. In the same year, also the hospital achieved Best Social Project for 2022, presented by Saudi Arabia and also by Project Management Institute. In 2003, the hospital also won Human Experience Guardians of Excellency Award by Breskin. So this is the hospital I came from the Holy City. So my agenda for today is I'm going to start with an introduction of quality control basic information. Then I will talk about importance of quality control for POCT devices, who's responsible for QC in POCT testing, choosing the appropriate QC procedure for POCT devices, choosing QC procedure for type of instrument, and I will end up by talking about QC material and external quality assurance. So quality is a really essential component of laboratory testing because clinicians rely on laboratory results for medical decision making, and any simple error will have a great impact on a great negative impact on diagnosis, management of the patient, as well as cost of care. So at the end, why quality control? It's an important detector for any errors that occur at the level of analyzer, reagent, operator, or the environment. And what we aim is actually to have a laboratory result that are correct and can be used for medical assessment of patient condition. Accuracy and precision are very important definitions in quality control, and accuracy basically means how close the numeric value to the true value, and it's indicated by the mean, while precision is how far apart the numeric values from each other, and it is reflected by standard deviation. So these are very important definitions that we know them very well because we use them on evaluating the performance of quality control. These are how statistical equations that we use to calculate the mean and standard deviation and coefficients of variation. As I said, it's also important in evaluating the monthly quality control. So our QC data actually range between plus and minus 2 standard deviation, where 95% of the QC data falls in between these 2 standard deviation. And as Dr. Kotb mentioned in details, these are Westgard rules that is used worldwide. And selecting which rules to use, basically it depends on what quality desire you're looking for and the level of method performance. So here, how do we do, in a simple way, how do we troubleshoot out of control QC? So you start by running the QC and you ask the famous question, is the QC acceptable or not acceptable? And if it is acceptable, you just report the patient result. And if it is not acceptable, you start to troubleshoot, looking for what could be the factor, right? So is it the reagent, is it the calibrator, is it the instrument, or the person who did the QC? Then it's a circle, so you repeat according to your finding, what you find, then you're going to start and rerun the QC again. Here I'm sharing with you clinical biochemistry lab, more sophisticated troubleshooting workflow that we use in the lab. In the point of care, it's a similar one, but it's a smaller. So you start again with running QC and you look for a QC problem, then the reagent, then the calibrator, then the operator, then the maintenance. According to your findings, you will do the corrective action. Then you rerun the QC and ask the famous question, is it acceptable or not acceptable? At this point, if you extensive all the factors, then you will ask for a help from a supervisor, maybe the company, to come and help you. So an ongoing assessment of quality control program, well, first of all, you have to set in what is your acceptability criteria, like the CV, the bias, and total allowable error, because these acceptability criteria is the one that you're going to use them to evaluate your monthly QC performance of your test. And therefore, you periodically evaluate the mean, the SD, and the CV. And you plan your corrective action according to your findings. And if you notice, or you should actually follow, if one QC for one certain test that is giving you a problem more frequently, you might want to compare the performance of that internal QC to your external QC performance. That will give you more hint and will help you in your troubleshooting. Challenging in point-of-care QC is mainly two things. First of all, it is user-dependent. So is the user, remember the user is not laboratorian, and therefore, is the user really, did the QC, did he know or she know that what to do as a corrective action for out of range? Did they actually document the QC? So it's about training. And the other things, QC in point of care is a bit expensive and costly, especially when we talk about low volume setting. So number of QC over patient ratio. So training, it's actually a collaborative approach between the POCT coordinator as well as the supplier. And you should include in your training, theoretical and practical, the storage of QC material, the preparation of QC material, appropriate frequency, principle and practice of QC testing, the troubleshooting and corrective action, and the documentation. And at the end, you want to also keep a proof of the assessment, either in a written questionnaire or maybe a simulation or observation. Who's responsible for QC in point of care? So if we start from the beginning at the end user, who performed it, he or she will perform the daily QC, and you will give them the proficiency testing sample to perform it as well. Now, the POCT coordinator will review the performance as well as the troubleshooting of QC and the PT report. Now, the medical director monitor the internal quality control and the proficiency testing program performance in general. Now, POCT committee will help you a lot because it's a multidisciplinary team that is involved, nursing and physician. They will understand more with you and the laboratory, of course, with you what's happening, and they can give an input on the decision and the discussion about a unit performance when doing POCT. Choosing a quality control material. Now, appropriate choosing, of course, you want the easier one, like use the liquid, ready-to-use quality control material that have a similar matrix to the patient and has a relevant clinical concentration. So if you are following certain cutoff, then you want the QC to be close to those cutoff or medical decision limit to test your method performance at that level. And also a third party, as Dr. Kotb mentioned, and she extensively talked about it, it's really good and recommended by many accreditation. Now, type of QC control material, we have assayed and non-assayed. The assayed control is actually have a specific value and range set up by the manufacturer. And for those ones, the lab is, the POCT section would have to verify the manufacturer claim. While non-assayed, it doesn't have a values or range set up by the manufacturer, and therefore the section has to establish the quality control values and the ranges for these. Both of them are very useful in taking decision for precision as well as accuracy performance of the test. Now, when choosing appropriate QC for POCT device, we have to look into two things, either the complexity of the testing or the type of POCT device. Now, for complexity of the testing, we looked at waved or non-waved test. So for CLIA and CAP requirement on frequency of quality control, for quantitative test, you need to have 2 control level at different concentration run daily of patient testing. Except for coagulation, then you need to have 2 level of control run every 8 hours. While for blood gas, you need to have at least 1 level of control run every 8 hours. Now, for quantitative test, you need to have a positive and a negative control each day of patient testing. For wave tests, the laboratory should follow the manufacturer instruction for quality control, review the result, and records acceptability prior to reporting patient result. For selecting QC for type of instrument, now it's divided, either it's high throughput instrument, you need a multi-level third-party control, should be tested minimum at least once a day on each day of patient sample tested. And if you're using cartridge-based instrument, there is a built-in QC designed to perform QC automatically without needing for operator interference. Now, the multi-level liquid form, how can I use it? Can I use it if there is a third? If there is a built-in QC? Yes, you can use it, and you will use it whenever you have a new batch, or to test the cartridge that is sitting on your shelf from time to time. The manual-based instrument, there are multi-level third-party QC on every day of patient testing, and some they have built-in positive and negative control. So here on the left side, you see the test cartridge where you have the chemistry reaction, and it contains the built-in internal quality control, while the device has the cartridge signals. It reads the cartridge signals, and it has an electronic check that tests and maintains the reader itself. So I'm bringing here to you an example for QC requirement per accreditation. So this is a daily QC requirement for blood gas instrument. So you need at least 1 level of quality control is analyzed at least every 8 hours when patient specimens are tested, and following the manufacturer instruction and laboratory procedure. So the lab has to define the number of QC level, the type of quality control used, and the frequency of testing. Okay? Now, it's also, whenever you resume patient testing, then you have to resume quality control with it. You have also to run QC whenever there is a major change that has happened within the instrument itself, like a major preventive maintenance, for example. Now, if the instrument has a built-in quality control, and electronic check, and you want to use those instead of the external quality liquid material. So at this point, to meet actually the quality control requirement, which is the 2 level of control on daily basis. So the laboratory must develop something called individualized quality control plan, and that has to be approved by the medical director or laboratory director, as well as you have to define the process exactly in terms of number of levels, as well as the type of controls, as well as the frequency. So here, what is individualized quality control plan? It's when the laboratory develop a quality control to balance between the built-in control as well as the liquid control, to reduce the frequency of liquid control, as well as to maximize the clinical outcome, available staff resources, and the cost effectiveness. Now, CAP has put in an algorithm for which test is eligible to do IQCP or not. But if I simplify it, here, if you are non-waved and you're not anatomical pathology, then the device has built-in QC. Then, if you want to go, the manufacturer, I can see it. Then, if you are not going to follow the your manufacturer recommendation is out of the CLIA requirement, then you are eligible to do IQCP. So it's basically you do a risk assessment and quality assessment, then you develop a quality control plan. That's what is IQCP. And this document, EP 23 from CLSI guidelines, is really very useful to build individualized quality control plan based on the manufacturer risk mitigation, regulatory and accreditation requirement, and individual healthcare and laboratory testing. So choosing external quality control assurance, now all regulatory and accreditation strongly recommend to use proficiency testing. And that's because it's very useful in assessing reliability of patient result, the accuracy of the result, as well as helping identify internal staff training needs. Okay, so this one here, just a CAP standard that require using of proficiency testing and external quality assessment. And if there is no proficiency testing samples for that test, then you have to do an alternative performance assessment. So the challenges in, I will talk about it in the next slide. Now, in external quality control challenges, there's a limited period between the test, when the time of the test is performed until the result you have. It is a low frequency testing. It comes like 3 or 4 cycles a year. And there is maybe an issue with the packaging. The samples actually came in glass vials. It might be broken. You receive the shipment late, or maybe it's not with the appropriate temperature. Some of the PT samples need to be reconstituted. So all these are difficulties. Others also include if you have unsuccessful test results, then you have to identify what is the cause, you troubleshoot, as well as you develop a corrective action plan, and of course recording all of that. Now, alternative performance assessment, as I said, it's actually used when external quality assurance is not available. And it is supposed to be done on regular periodic time, like semi-annually. One of the examples is to do split testing with another laboratory, or you compare your result with another method within the lab. The advantage of it that we use patient samples, and you are actually testing the pre-analytical procedure within it. Now, the limitation of it, the uncertainty of, or the differences between the two methods that you're comparing yourself with, You cannot assume that your result is the correct result, and there is no peer to compare your result to, and the sample could be deteriorated during the transportation. So I'll conclude with here that quality control management of POCT instrument is challenging. Principle of internal and external quality control should be applied, but there is no universal strategy. Principle of evaluating and troubleshooting QC is comparable between the lab and POCT because errors may arise from the same factors, which is the test system, environment, and the operator. And thank you.
Thank you, Dr. Heba. We will now continue with Dr. Heba's insightful presentation on future of point-of-care testing, which is due to schedule constraints. We will have to bring forward a day earlier for your benefit. Please, Dr. Heba.
Thank you. So I was asked to talk about POCT in the future, but I cannot talk about the future without talking about the past and the present. So I'll start by bringing some history, and then I'll talk about the importance of POCT applications, then the eyes on the futures, and thoughts about POCT. So as President Theodore mentioned, the more you know about the past, the better prepared you are for the future. And we cannot mention the history without talking about the great Egyptian civilization that lasts over 7,000 years. And it's a great civilization, and the technology was huge at that time, and it impressed us as we know and study about about the Egyptian civilization. So at that time, a pregnant woman were asked to pee on wheat and barley, and if the wheat grows, that means she is pregnant with a girl, and if the barley grows, it means that she's pregnant with a boy. And second, we have the Byzantine Empire, which is called the vanished world. And the reason for that, because it's been replaced totally by the Roman Empire as well as the Ottoman Empire at that time. And what is the practice there, patients were asked to be given a urine sample where they compared to different sample of urine held on a urine wheel to help on making a diagnosis at that time. So 20 years ago, we brought the bedside testing as a project, and we started by what we call it point of care. An early definition is a test done by non-laboratory staff outside the recognized diagnostic laboratory, and it has a couple of terminology that's changed over the year, which is near patient testing, bedside testing, extra laboratory testing, disseminated laboratory testing, and finally point of care testing. Here are some definitions from regulatory and accreditation bodies like CAP, ISO, and CBAHI. And pretty much all of them, they were having the same definition in terms of the test design to be used near to the patient and is located outside the lab and doesn't have a permanent dedicated space. And it can be outside the physical facility of the clinical laboratory. So changing point of care changing concept, we started by having a reliability of result where quality control and competency are very important rules. Then we moved to patient-centered care where patient safety, quality of care, and faster clinical decision are very important. And finally, right now, we are at optimal patient care, where patient satisfaction and physician efficiency are very important. So when having a point of care testing or thinking of developing it, you might want to think about complexity of the testing, the location where it's going to be, and who's going to do it, and what we are providing as turnaround time. Now, what we have here is how we managed to develop or adopt a QC, a point of care, in our hospital. So first we start by looking at the clinical need, linking the evidence with the need. Then we look at laboratory review and approval. Then we do, of course, the method evaluation and the whole process evaluation. Then we implementing it and monitoring it. So quality result recipe, which is accurate, precise, and reliable and timely result. And that recipe should be all concentrating on optimal patient care by having a quality management program, effective training program, effective competency assessment, continuous monitoring, and continuous improvement. Now, home versus hospital, and the famous one is glucose meter. There's a difference between them? Yes, there is a difference between them. Like, for example, home meter is used by one person, while the hospital meter used by a couple of them. The performance is less in home meter compared to the hospital. The detection, even the specification, is narrower in the home meter compared to the hospital. And of course, the connectivity may be available in the hospital, not yet available for the home glucose meter. And do we need that? That's a good question. So where did we achieve in POCT connectivity progression? So we started by manual paper records. Then POCT devices are directly connected to the computer. Then manually entered result in the hospital information system. Then the POCT devices are connected to a middleware solution. And finally, we have a wireless connectivity. So on here, I'm just going to mention quickly the POCT test menu is actually expanding from time to time. And the list is huge in chemistry, as you see, compared to hematology, but it also includes coagulation, toxicology and therapeutic drug monitor, as well as infectious disease, and finally molecular. So the technology is there, and our eyes is on the future. What are we expecting for the future? So the technology and the companies impress us every day with new devices that come in, like the smart contact lens for monitoring blood sugar for patients with diabetes. But is that all what we want? Or we want to have a better experience for our patient? And also you heard about the smart watches that can measure your heart rate and directly connected to emergency ambulance and stuff to come and help the patient whenever there is an emergency with the patient. Now, so this connectivity, that's what we wanted, right? So I believe the future is in telemedicine, where we can actually, with virtual care and use of point of care testing, we can help physicians to triage those patients that really needs direct admission through fast track from the emergency. You get the point? So from home, triaging them through telemedicine to the direct admission to the hospital. I believe with this, we can really reduce number of emergency visits. We can reduce the complication that can happen to patient because we are actually detecting them earlier. We can improve physician in their efficiency. And hopefully with all of this, we will help to reduce average length of stay in the hospital. And finally, we'll improve patient experience. This is a publication that would put in, it was about POCT quality assurance program requirement to ensure producing reliable results and patient safety when using POCT devices outside the hospital setting. It is very nice and good document published by POCT committee from the IFCC. I'm one of the authors, so if you want to read it, it's really useful. So I will end up by here saying that technology can impress us, but I believe the experience that we are creating with this technology, that is the one that will make a difference. And that's it. Thank you.
Thank you, Dr. Heba. We will now open the floor for questions. I'd just like to remind you, if you'd like to ask any question, you can scan the QR code and submit your questions online. So we have a few questions. The first question, I think, is for Dr. Thana. The question says, on changing lot number of control, is it mandatory to reassess Six Sigma? And just to continue on that, the next question is, how many times should we calculate Six Sigma?
Regarding Six Sigma, it is changeable with time. The recommendations is every 6 months you can change your Sigma. You can look at, because you will increase the CV of the assay with previous points. So it is not, less than 6 months, it will be very difficult to apply and to go on.
Thank you, Dr. Thana. The next question we have is, can we deal with 2 or more labs using different instruments to check the same samples monthly and consider it as external QC for values and references used? Should I repeat it?
That's for point of care, or that's?
The microphone's working, yeah.
Okay. So using an alternative assessment, is that the question?
Yes.
Okay. So yes, split testing is one of the alternative assessment that you can use it while you run a specimen within your lab and another lab, then you use it, you compare it to your acceptability criteria. Yes.
Thank you. The next question is, I think this is basically what you mentioned. How can we suspect light source degradation or degradation on an instrument measuring is gradually weakening from the Westgard curve? How can we suspect some kind of deterioration in the light source from the Westgard curve? I think they mean the leaving Jennings.
As we said, some of the rules are sensitive to random error. So they are mainly focusing on imprecision. And some rules, they are more sensitive to systematic error. So systematic error, usually we focus on the reagent, the calibration, and most of the systematic error are related to calibration process, which were instability of the calibrator or reconstitution error of the calibrator or using a new lot number of reagent or calibrator. Also, it can be in the QC material. But mostly the random error, you have to focus on the instrument itself. Sometimes instrument issues can lead to systematic error, which is having the same error affecting all the patient samples. For example, deterioration in the photometer or in the lamp can affect the runs with the same value, so it can lead to systematic error. So it is not, as we said, Quran. You have to also think and have brainstorming when you have a rule violation, and so that you consume the time, not to spend the time for fixing something which is not the cause of the error.
Sorry, I have a question to Dr. Heba, please. Comparing biochemistry analyzer in the laboratory. I have, for example, 3 biochemistry analyzer. And in some hospital, they may have 200 point of care for biochemistry in each ward, and in each casualty, many. Some of them, I'm not exaggerate, maybe they have even more than 200. So to say it is easy to run the internal quality control, external quality proficiency testing for the 3 machines in the lab. So how can I will do? I have now 200 machines. How can I will manage? You understand my question? Yes.
Now, just to make it easy, if the device has a built-in QC, so that will help you a lot in reducing the cost of running those QC by developing that IQCP program. This is one thing. The other thing, in terms of meeting the PT requirement for accreditation, what CAP nicely have done recently, they have something called, a program called a cross-check, where you use one quality control for multiple instruments at the same time, and you will get the same result, and that will help you really know the performance and compare them between those devices. Because at the end, you don't want to have one device giving you wrong result and people are relying on it while the others are okay.
Patient sample, not the quality control sample.
No, it is within the program itself. Like CAP provide those specimens for multiple, like in one survey you will have multiple same sample to be run on all these devices. So you will enter, for example, for CAP, that you have that number of devices and they will provide you. You get my...
Yeah, but it's if I have 200 instruments.
Yeah, you have to run quality. Because the reason here, as I said, that you cannot just leave one device that is not, you're not sure of the performance of that device. Because people are making decision on it.
I have comment here, Dr. Heba, because we have the same challenge. We have to differentiate between point of care, which is a reading device and all the reagents, a calculator, everything in the cartridge, unlike the point of care, which is like blood gas analyzer, where all the reagents and the calibrator are inside the machine. So this is our challenge also when we have the iStat and we want to run the control material in it. So all the changes are in the cartridge, not in the reading device. So we tried to also, like Dr. Heba told, but this was our internal control, to bring in the same site the number of iStats they have and divide the QC material, because it is ampoule, it has to be done at one time. Yeah. So we distribute on some of the cartridge on the 4 or 5 equipment in this location. Next day, we will do the other location. And we will do also when we receive the cartridge, so that to be sure that no transportation error for the cartridge we received. And we repeat again when we have any discrepancy between the lab testing and the point of care, and between the point of care and the clinical decision making or the clinical picture of the patient. So we were doing what's called individualized QC plan, just to save out. But this was all about internal QC. So about what Dr. Heba is talking about is external QC. The CAB is giving, this is, I don't know because we don't share an external quality control program for the point of care till now, but this is mean that CAB is giving you a number of samples that covers all your point of care. So it's about enrolling in the program in the beginning.
No.
The number of instruments you have, you will enroll in.
Doctor, we said the built-in. If it is the instrument, there is a built-in QC.
Yes.
It will be enough?
No, that will be... The
Yeah.
built-in
Yeah.
QC is for internal quality control, not
Okay.
for the external quality
You want
control.
again an external?
Yeah, you have to do external as well.
It will be too much.
That's a must. Just to make sure that all the devices are performing well as the rest, as each other.
Dr. Heba, if I can add to what you're talking about, one of the questions is, what are the chances of error in a point of care testing result, and that will affect treatment? So I think their question is about inaccurate results.
Oh, it's per test. Like it's variable between tests. Like glucose is different. The bias in glucose versus troponin versus blood gas is different. And Westgard has really a total allowable error list of all the majority of the testings there.
Okay.
Sorry, Doctor, I'm just argue with you about point of care. No, because of course we are thinking about cost. If I have this number, if I, for example, point of care in Kuwait, it will be hundreds, even thousands. So I can do my own, for example, like I have a pooled sample, I can run it, like external, local external, because I am sure if I am doing this external, which is outside Kuwait, it will be much expensive. But if I do a local one, where I do a pooled sample, then I will run it for my hospital. What do you think about this idea?
Well, it depends if this is acceptable by the accreditation your laboratory has. If the accreditation will accept that. Like for example, if we're talking about CAP, they will not accept it. As long as they have proficiency testing provided for the test, then you have to use it.
Thank you, Dr. Heba, and thank you to all our participants today for your questions. I'd like to invite the speakers just to receive your certificate of participation.